The Bohr effect is when changing in pH will affect hemoglobin's preference for T state or R state, okay? And we saw that as a function of pH, essentially lower pH prefers to have the T state, higher pH will then prefer having R state. And then the reason being that at lower pH, your histidine 146 is protonated. So if histidine is protonated, so protonated histidine is what charge? Do you guys remember? Histidine, if it's protonated, is it neutral or positive? I mean, obviously protonated, you will not be minus, right? Right? 听懂哈? Protonated, meaning you have a proton on the molecule. 它就不太会是 minus, okay? 它一定是 neutral or positive. For histidine, it's going to be positive, okay? So protonated histidine is positive. And because if you protonate histidine 146, it's going to have a very nice salt bridge between aspartate 94. And here remember that aspartate, this guy, is an acid. Acid at normal pH, it's always going to be minus. Okay, so for your purpose and my purpose, glutamate and aspartate, okay, so GLU and ASP, those two guys, they are always minus for our purpose. They're always negative. Why? The pKa is like what? pKa for the R group is 4. Okay, so they have that, you know, carboxylic acid as their R group. And then the pKa for the R group is what? 4. Okay? And physiological pH is like how much? Like 7, something like that. All right? So if pH is higher than the pKa, it means it gets deprotonated. Okay? So actually, for you guys, seriously, if anything is important, being able to tell whether or not your thing will be protonated or deprotonated is probably like the most important thing in this exam. So aspartate at normal physiological pH is always minus. So you have a positive, minus, they form a very good salt bridge. Okay, a salt bridge. And because it forms a very good salt bridge, it stabilizes the T state. Okay, remember that because in R state, the association of the residues that can form salt bridge gets too far apart. So R state has no like salt bridge, okay? But T state, you do have salt bridge. These residues are close to each other. And then with a lower pH, your histidine becomes positive, okay? Because normally it's neutral, okay? And then it becomes positive, and then what you have is a good salt bridge formation. Proton affinity is also higher in the T form. Why? Because at the T state, your histidine is going to be close to your aspartate. So where's the histidine? This guy is the histidine. This guy is the aspartate. So only under T state, aspartic acid residue, so this guy, will get really close to the histidine. Okay, and we just said that aspartic acid is minus. Okay, and having a minus there, it will actually increase the pKa of the histidine. Okay, why do you want to increase the pKa of the histidine? Think about it. So let's say the pH is at 7. A typical imidazole pKa, so this is normal histidine, okay, and then this is, you know, hemoglobin, T state, histidine. And then for the normal histidine, if it's just normal histidine, it's 6.4. If it's in the T state histidine, it's 7.9. And by T state means that aspartate is close. Bye. So we know that you have two different pKa. So at pH 7, let's say we're at pH 7, the normal imidazole is going to be protonated or deprotonated? deprotonated, right? So this guy is going to be deprotonated. Here it's going to be protonated because one is the pH higher than pKa, the other one is pH lower than the pKa. Okay, so if it's deprotonated, it's neutral. If it's protonated, it's positive. Okay, so actually having that aspartic acid residue there actually makes your histidine want to be protonated so that they can form a good salt bridge. So the presence of that aspartic acid there actually promotes this process. All right, another thing that hemoglobin can do is that it can carry CO2. So what does hemoglobin do? Hemoglobin carries oxygen around and it circulates in your body. In addition, what it can do is it can bring CO2 from the tissue back to the lungs. So a small portion of the CO2, estimated to be about 5 to 13%, reacts directly with hemoglobin. Okay? How does it do that? The N-terminus amino group of your hemoglobin is going to be positively charged. And what that will do is, remember here, CO2 is in equilibrium with bicarbonate. Bicarbonate will react, this guy, react with your amino group, and then you can form this thing called carbamate. Okay, so that's what this thing is. So this thing is a carbamate, and this carbamation reaction will allow hemoglobin to aid the transport of CO2 from the tissues to the lung or gill. If you're a fish, you have gills. If you're an animal, you have lung. And this process, the proton here is released through this carbamation reaction, and this also then decreases pH and then causes your Bohr effect. So, Bohr effect. In summary, what we have here. Again, this curve here, that's the myoglobin. And remember, myoglobin has no cooperativity. So myoglobin has always the same affinity for O2, regardless. Now, hemoglobin, remember that it has cooperativity. So it changes affinity depending on the amount of O2. Now, what we see is that as a function of decreasing pH, so you go from 7.6 to 7.2 to 6.8, what you see is that you have a decreasing O2 affinity. What that means is that it actually helps your hemoglobin to release O2. Remember, we say that around the tissues, right, around your tissues, you have higher amount of CO2, and having higher amount of CO2, you have more proton, so lower pH. So that makes sense. And even it also makes sense is that when you exercise, you also produce lactate. Lactic acid also produces you a lot of proton, right? So at tissue, at your like hands and stuff, at your tissues, you have a lower pH than other places in your body because you have very good respiration going on, you generate CO2, okay? And as a result, lower pH will cause a preference to go to the T state, the deoxyhemoglobin. Okay, the T state is stabilized. So if the T state is preferred, T state is preferred, then the graph, your O2 binding will look like that. Okay? Because here T state is preferred. Remember, T state does not like to bind O2. R state likes. So this is how CO2 and proton can affect your hemoglobin. So CO2 also increases the proton around your tissue, and then your tissue also generates protons because it may produce lactate and things like that. And then as a result, your proton will cause Bohr effect, which essentially is saying your T state will be stabilized. Okay? If T state is stabilized, then hemoglobin does not like to bind O2. Therefore, it releases O2. All right. Another thing that also helps Bohr effect is chloride anion, okay, Cl minus. So in the T state here, you have valine 1, okay, 1 meaning this is first amino acid. We know valine is a hydrophobic amino acid, right? So you're like, what? Valine doesn't do shit, right? But no, because it's the first amino acid, it has that N-terminus. Okay? And in the T state, your valine 1 is right here. Valine 1 is close to arginine 141, which is this guy. They're close to each other. And what happens is that the chloride ion, Cl- ion here, it will actually promote the protonation of your amino hydrogen on valine 1. Okay, so here, this is NH2, right? So having this minus charge here, it's going to promote your protonation of a proton on your N-terminus, and then here is going to be a positive charge. And then that's also helping the Bohr effect. So Bohr effect is the formation of your salt bridge as a function of lowering pH, right? So it's going to help you to have a stabilization of the T state, okay? So Cl- also can contribute to stabilization of T state, which then can help you release oxygen. Why doesn't it react with R state? That is because the valine 1 and arginine 141, they're too far apart in R state. Okay, so only in T state you have salt bridge going on. Okay, so that's Cl-. So another thing, so we talked about proton, CO2, Cl-. The other thing that I want to talk about is this thing called 2,3-bisphosphoglycerate. So 2,3, meaning here is 1, 2, 3 carbon. Bisphospho, meaning it has 2 phosphates. Glycerate, this molecule is glycerate without the phosphates. So 2,3-BPG is a compound that's found in mammals. In birds and stuff like that, you have this thing called IPP, which is myo-inositol 1, 3, 4, 5, 6 pentaphosphate. So it has 5 phosphate groups. Now, what happens is that 2,3-BPG is found inside the red blood cells, and it's a potent allostery effector that lowers the affinity of hemoglobin for O2 by preferential stabilization of T state. So this thing likes T state. Okay? If it likes T state, what does it do? Okay? It lower O2 affinity. Okay, so it works together with the Bohr effect. Okay, so everything we talked about all prefer T state. Everybody want to make it happy for T state. All right? So let me show you how it works. Turns out 2,3-BPG is negatively charged. I mean, no shit. You have 2 phosphate, okay? 2 minus, 2 minus. So total 4 minus. So this thing is very negatively charged, so it will form very good salt bridge with positive charged hemoglobin at the center. Okay, so at the center of hemoglobin, remember we said that the 2 alpha and 2 beta, it will have a center cavity. There's a hole in the middle of the hemoglobin. At the center, there are quite many residues that are positively charged. For example, you have the N-terminus amino group. You have histidine, okay? You have lysine, histidine, lysine. So you got a bunch of these positive guys. So if you have 2,3-BPG, which is 4 minus charges here, okay? So you got a lot of minus charge. So you have a lot of negative charge. Okay, so if this negative charge goes into the center of this molecule, this hemoglobin, then your hemoglobin will all of a sudden have a lot of salt bridges. Salt bridge, remember, stabilizes your protein. Okay? So this is stable. Okay? I only write it so big because it's important. Now, 2,3-BPG only interact with the T form. Why? Because the hole, the center hole for T state is larger. R state is smaller, okay? Because it's smaller, then your 2,3-BPG doesn't go in. So that's why 2,3-BPG prefer T state. Once T state has 2,3-BPG, once it's stabilized, then this guy does not want to go back to R form. In most of your tissues in your body, if you have 2,3-BPG, nice stuff, okay? Good stuff. Now, One thing I'd like to share with you guys is that during developmental stage, your hemoglobin actually change. Okay, so when you're a fetus, like a little baby but inside of your mom, okay, when you're a fetus, then you don't have alpha beta for your hemoglobin. Actually, you have alpha gamma. So this is adult hemoglobin shown in this picture. This is fetal hemoglobin shown in this picture. Now, what do you see different? What is different is actually on the beta subunit. So you don't have the beta subunit, you have gamma subunit. And what's different? Gamma subunit does not have this histidine. This histidine normally will give you a positive charge which interacts with your 2,3-BPG and T state. It stabilizes the T state. Now, for fetus, like unborn baby, for fetus, you don't have histidine there. What you have is a serine. Here you have an OH group instead of imidazole. So this guy, regardless, is neutral. It's neutral. So what does that mean? That means that it binds O2 stronger because it has a lower 2,3-BPG effect. So adult 2,3-BPG have a higher effect for T state because you have histidine positive right here, and where's the other one? This one. Okay, so for adults, you have histidine over there, which is positively charged, which helps the 2,3-BPG bind there and stabilize the T state. Okay, so T state super stable in adults with 2,3-BPG. For babies, I mean fetus, for fetus, you have gamma subunit instead of beta subunit for hemoglobin, and you don't have that histidine. You have serine or threonine, I forgot. Okay, but you have a hydroxyl group there, which is not charged, so no charge. No positive charge. So you lose two salt bridges that stabilize your T state. Okay? So your T state is less stable compared to the adult phase when you're a fetus. Why is that a good thing? Why is that a good thing? Okay? It's actually in my next slide. But think about this. Okay? If you are a fetus inside of your mom, okay, what do you need to do? You need to compete for oxygen, right? So you need oxygen. So then if you, the little fetus and the mother have the same hemoglobin, then you have no advantage in competing for O2. So now here you have a preference for fetus. You have a preference for the R state, right? Because adults have more stable T state. Fetus? No. Okay? Fetus does not. So therefore, fetus prefer R state, which then grabs O2 better. So if you look at birth, this is birth. So before you're born, inside of the mother's body, primarily you have gamma over beta. But then after you're born, after the baby is born, gradually you have beta and then no more gamma. So turns out the Bohr effect with the CO2 and the BPG, they can have a cumulative effect. So this was an experiment done with hemoglobin stripped out of CO2 and BPG. So this is stripped hemoglobin, so it's just hemoglobin. And you can see that it has a very good affinity for oxygen. Now, when you start adding either CO2 or BPG, like this line right here, is you add CO2, you see how the affinity, let's say we draw a line maybe like right here. For example, you see how the affinity drops. I'm going to use a different color. So if you draw a line like right here, you go from here to here, you dropped affinity. Okay, so here it says that essentially if you have CO2 together with the hemoglobin, affinity will decrease because you prefer T state over R state. Remember? Now if you add BPG, it drops even more. The effect of BPG is higher than the effect of CO2. Why is that? Because the salt bridge at the middle. Salt bridge is super important because if you have salt bridge, your protein is super stable. Now, what you can show is if you add both hemoglobin and BPG and CO2, it's even more stable. And that's actually equivalent to what you observe in the blood. Okay, so here inside of your blood, essentially it tells you that in your blood you do have a lot of the Bohr effect and 2,3-BPG that stabilizes the T state. 2,3-BPG and CO2 stabilizes the T state, which lowers the affinity of your hemoglobin for oxygen at places that don't have a lot of O2. So here, like low oxygen, you want to have a low affinity for O2. So what you see is that at low oxygen concentration, you have affinity that gives you about 30% or 40% saturation. Okay? Whereas using the same curve, at high level of O2 concentration, you get about 90%. So you have a huge window for delivering oxygen to your myoglobin. So that's the biochemistry aspect. So now I'm going to show you some stuff related to genetics. This is eukaryotics, like us, like we have hemoglobin, so we're eukaryotic systems. We have exon, intron, and stuff like that. So this is how your hemoglobin is made. This is beta globin. So you got DNA. DNA gets transcribed. You have a premature, a pre-mRNA, and then you have mRNA, and then you have translation. Now, what I want to highlight is that current understanding is that mutations generate diversity, and diversity allows for evolution. So diversity gives rise to advantageous traits that enable an organism to survive. So for example, if a protein is needed in large amount and is important, then a gene duplication may occur because having more of this protein is good for survival. Then selection, okay, Darwin's theory there, selection will prefer and select for increasing amount of that gene. Here I want to give you some terms. Homologs. Homologs are proteins that are related by common evolutionary origin. These proteins tend to have similar structures and sequence. Homologs may or may not have the same function, so they look the same, but they may or may not have the same catalytic function. That's homolog. Now, among homologs, you have two different kinds. You have paralog. Paralog is homologous gene within an organism that arise from gene duplication. Yeah, so for example, like humans, we have multiple alcohol dehydrogenases that may evolved as gene duplication, for example. Okay? Ortholog, on the other hand, are homologous genes in different species that originated from a common ancestor. For example, you and monkeys, you have similar genes, right? But you guys are not the same organism. You're two different organisms, but you share similar protein. Those proteins are often referred to as orthologs because they evolve from a common ancestor. Why is this important? Because we want to show you the evolution of globins, okay? Now, you have some kind of ancestral globin, or globin, depending on how you want to say it. So, through evolution, you split and you can have neuroglobin or hemoglobin, and hemoglobin have its alpha chain, beta chain, even though they are different polypeptides, but they have very similar traits, right? They both have heme, they both carry O2. Now, you can also evolve into some other proteins, for example, myoglobin, right? So myoglobin, very similar to hemoglobin, or you have cytoglobin, okay? And this is sort of a gene duplication event that allowed you to have different globin genes on different chromosomes, just to show you that they're all related. Right? Biochemistry is not that complicated because what you'll see repeatedly is that many genes, they come from the same origin. Okay, now, this is something you should have covered back in general biology about mutations in DNA, You have, for example, here is the beta gene for hemoglobin. This is the normal gene. You can have 4 types of mutation that can occur. One is silent mutation, meaning that it does not change the protein, okay? It doesn't change amino acid. In this case, it's just a silent mutation like this. C become T, even though it's a DNA mutation, but histidine remains the same. That's a silent mutation. Or you can have sense mutation, which occurs that changes an amino acid. Okay, for example, you change this A to a T, then you go from glutamate to valine. you change the structure of your protein. That's called missense. Nonsense is where you change one amino acid and it leads to a stop codon. It chops your protein prematurely. Okay, then if you have a nonsense mutation, the original protein may have 400 amino acids. Now the mutated protein has 100. Okay, it has a shorter protein. Or you can have a frame shift. Frame shift sucks because you either insert or you delete. You will cause a frame shift. As a result, you have a super messed up protein. Okay, so these are the 4 kinds of mutations that DNA could have. Now, why am I mentioning this? Because hemoglobin has been or have been involved in many diseases in humans. Okay, for example, sickle cell anemia. Sickle cell anemia is caused by a mutation of that glutamate to valine, and we'll see that in just a minute. And you can have other effects, for example, change in O2 affinity. That's when you have altered amino acids. Okay, always you have some kind of amino acid change, and then you have decreased function or a mutated function or whatever. Now, related to hemoglobin, you have cell sickling, you have change in O2 affinity, you can have heme loss. Heme loss is very serious because if you lose the heme, your protein is dead. Okay, your hemoglobin is dead. Okay, it's not even good. Or you have dissociation of tetramer. That's kind of sad because then the thing kind of becomes like a myoglobin. The one that I want to highlight, of course, is sickle cell anemia. This happens a lot, especially in some part of the world, especially in Western Africa. What you have is a mutation, a missense mutation that turns a glutamate into valine. Okay, you're just like, what kind of thing it would do to me, right? So it turns out, valine just so happens that it provides your subunit with a super nice shape, like this, super nice shape. That allows it to sort of interact with another subunit of the hemoglobin such that they form a very, very stable complex. So essentially you can think of it as a lock and a key mechanism. So having that valine here, you have a very nice fit between subunit 1 and subunit 2. And then what this does is it actually causes a polymerization of your hemoglobin. Okay, so the normal hemoglobin looks like this. Okay, so it's like round and stuff like that. It's very nice. The sickle cell hemoglobin has a cavity, has a key. Okay, so it looks really ugly, but you get the point. Okay, so what you have between the sickle cell hemoglobin is that they can form very nice linkages. This one will go with another one, okay? So that you can form polymers. You start to form like a little filament, okay? And that's a problem because your red blood cells, hemoglobin is primarily the protein inside of your red blood cells, right? Your blood vessels is flexible, is thin, okay? And you want your red blood cells to be also flexible so that they can pass through many different places, okay? Now, if you have that valine mutation, your hemoglobin now forms polymer. Then what it does is it forms this large crystallized structure that causes your erythrocytes or the red blood cell to have an elongated shape, okay? And this guy is no longer flexible. And as a result, you will have anemia. Anemia 就是缺血嘛, okay? Anemia. Right? So some of the symptoms, you have anemia, pain, swollenness, frequent infection, vision problems, delayed growth. So this is a hereditary disease. Okay? It happens on your DNA. Okay?